Okay, so we're gonna talk about late part of DCT and cortical collecting tubule functions. So basically, this is a late part of DCT, and collecting tubule has two parts, one which is in the cortex and one it is in medulla, right? So late part of DCT and cortical collecting tubule has almost same function, okay, because they have similar cells. They have principal cells and intercalated cells intercalated between principal cells. So intercalated cells are acid secreting cells. So what they have, they are very rich in carbonic anhydrase enzyme. So carbon dioxide water combines with in the presence of by this enzyme forms carbonic acid, which dissociates into carbonate and H plus. This H plus is excreted out into the lumen by uh, hydrogen ATPases, right? They actively secrete out this hydrogen ions with at very high concentration, right? Because they are very much efficient that they can excrete out hydrogen into the lumen, which is 4 times highly concentrated with H plus. Okay, they are very much efficient ones. And then what happens? Okay, so they release H plus into the lumen, and this H plus is actually taken up by this phosphate and ammonia, which are filtered from the blood, right? So these compounds like HPO4 and NH3, they take up this H plus and they form H2PO4 and ammonium chloride. This is basically helpful in this actually traps up highly reactive proton ion, and they also decrease the pH of, they also does not allow the very much increase in the pH of acid, right? They they slightly make it quite less acidic, okay, as proton is trapped in them. And this carbonate is reabsorbed, and which can be reabsorbed with the potassium also, potassium carbonate, right? So what happens in acidosis is basically there is increased acid in the body, so this carbonic anhydrase is strongly stimulated, so it produces a lot of HCO3. And this same mechanism happens like HCO3 is combined with the H plus to form carbonic acid, and this carbon dioxide can be expelled out as there is increased rate of respiration, which happens in few cells. And at the same time, what happens is this mechanism is also hyperactivated, where this carbon dioxide plus water, we should know that this carbonic anhydrase enzyme is a it can work in the bidirectional way, okay? It can favor this, it can also form this, okay? It's a bidirectional enzyme, by the way. Okay, so in case of acidosis, this enzyme is stimulated in the cells, which results in formation of excess of H plus, and this excess of H plus are excreted in urine. So there is in acidosis we find increased level of HTPO4 in urine and decreased levels of HPO4. At the same time, ammonium chloride levels are also increased in urine, okay? So in acidosis, two things happen. This also results in the formation of carbon dioxide so that it can be expelled out through respiration, and there is increased acid excretion through urine in case of acidosis. Now let's look at the principal cells, what they do. They have a sodium potassium ATPases on the basolateral side, which take out sodium out and potassium in, making the cell sodium poor, right? And this cell also have few channels called sodium potassium ATPases. Right. So this cells make these channels make the cell sodium poor, sodium potassium ATPases, and it has sodium channels and potassium channels on luminal site. So when the cell decreases in the concentration of sodium, this favors movement of sodium from the luminal fluid into the cell. In this way, sodium is reabsorbed, and as potassium concentration is increased in the cell, this results in excretion of potassium, and potassium is excreted into the urine, okay? So there is an important hormone called aldosterone, which is produced by the zona glomerulosa of adrenal cortex. This basically aggravates the whole system, the whole process. It activates the genes and increases the synthesis of sodium potassium ATPases on the basolateral membrane, and then inserted on the basolateral membrane. So what happens is more sodium leaves the cell and more potassium enters the cell. This makes the cell extremely sodium poor, so more sodium is absorbed from the lumen into the cell, right? And at the same time it increases the potassium channel synthesized, so as more potassium entering and more potassium leaves out, causing calcuresis, right? More potassium is released out into urine, and at the same time it also increases the concentration of sodium channels, so that as more sodium is being taken up by the sodium potassium ATPases into the lumen, cell is becoming more sodium poor and more sodium can be reabsorbed. So basically what this hormone does is it results in increased sodium and water retention, right? Sodium will always drag the water with it, right? To increase sodium and water retention and potassium loss in urine, so this is responsible for increasing the volume of blood. So this hormone is released in case of chronic heart failure, congestive heart failure and shock, hypovolemic shock, and Cone syndrome, in which there is a tumor of zona glomerulosa producing excess aldosterone. So we should know this, as per sodium reabsorption, 65% is reabsorbed in PCT. Leukophenly has 25% reabsorption in the ascending part of leukophenly, and 5 to 10% in DCT, and 3 to 7% in collecting tubule. And so this 3 to 7% is regulated by this hormone aldosterone. Alright.